Hello children, I am Dr Aruna Mohan from Delhi University, and today we are going to discuss about dihybrid cross. Before that, I would like to remind you that in the previous session or previous episode we discussed about monohybrid cross, and we discussed the quality of dominance and recessive natures. Before I go to dihybrid, I would like you to know a few things which do not follow the rules exactly, but do follow rules eventually. One good example in this is incomplete dominance. In the monohybrid cross, when we were crossing tall plants with short plants, of course, pure tall with pure dwarf or pure short, in F1 generation we got all the tall because tall is dominating character indicating law of dominance. But now, if we take example of dog flower, it can be of red color, it can be of white color, and you may have seen pink color dog flowers also. Now, if we cross pure red dog flower with pure white dog flower, then if red is dominating, then F1 generation should be all red, and then the next generation should indicate 1:2:1. That means the F1 is going to be 3:1, but in case of dog flower that will not happen. You will get all pink flowers, that is a color between red and white. That means the dominating character red, which should have shown itself, is not showing fully, but showing partly, that is why it is pink. Now, this is called law of incomplete dominance. The dominance is not full, but when you cross pink with pink, that is F1 generation crossed with each other, then in F2 you are getting, you can see red, pink and white in the ratio 1:2:1. That means F2 generation is following Mendel's law fully, like it happened in pea plant. That means both the characters were either hidden in F1 or it was incomplete dominance. So, in genetics and heredity, children, this can happen that some of the things will not follow the rules exactly, may follow it indirectly, and this is one example, and we call it incomplete dominance. Similarly, there is one more example which I would like to discuss before I go to dihybrid cross. It is called codominance. As the name indicates that both the characters will be shown in the same individual. A very good example for us is our blood groups. Children, you know that our blood groups are A, B, AB and O. So, most of us will fall in one of these 4 blood groups. But when you see the genetics of it, the blood group is governed by a gene which has 3 alleles, say allele A, allele B and I, small I. Now, your blood group is decided by 2 of them. It may be A and I, then blood group is A. It can be B and I, then blood group is B. It can be AB, then blood group is AB. It can be II, then blood group will be O. That means this is codominance. Both the characters are present and whatever is present is showing. I think better example, easier example for you will be cow. In some cows you will see there are patches, brown patch, white patch, again brown patch, again white patch. Now, for this cow the parents were, one was brown, one was white. Then if it was incomplete dominance, then color between brown and white, something like light brown or gray kind would have come, but it is not the case of that kind of dominance. And if it was a case of law of dominance, then out of 2 one should have come, either brown or white, whatever is dominant. But both are coming, that is a case of codominance, that some part will come from male parent, some will come from female parent, and they will show itself exactly the way it is shown in parent. So, it is the example of codominance. Now, after understanding these two kinds of dominances, that is codominance and incomplete dominance, now we go for dihybrid cross. Mendel conducted dihybrid cross to see the results. He wanted to prove that his result is correct in monohybrid also, in dihybrid, and in all the characters in our body. So, what he did? He took 2 characters. These are the Mendel experiment I am trying to tell you. He took round seeds and wrinkled seeds, and he took green seeds and yellow seeds. So, he took 2 contrasting characters from the seeds only. As he had been doing, he made pure green seed, pure yellow seed, then pure wrinkled seed and pure round seed. After making pure plants, he tried to cross pure green and pure yellow seed with wrinkled and green. Let me repeat, round seed is dominant and wrinkled is recessive. Similarly, green color here, green seed is dominant and yellow is recessive. That means, we are going to show green with capital R, and we are going to show the recessive character with small r, and similarly yellow and green. Now, after he crossed these 2 characters, naturally the result was interesting. He will get all dominating character, that means he will get round seeds and he will get green pods, and it will follow the same Mendelian laws. If you want to conduct this experiment, this is what Mendel did. Now, I am going into detail of this experiment, so that you can also conduct it in your lab or in your class. You take 4 colors beads. First you collect beads and then take 4 color beads in 4 different beakers. You can choose any color. Of course, I am going to explain you with 4 definite colors, but you can decide any color and accordingly put them in beakers. So, I have separated 4 color beads in 4 beakers. Now, you count and take 48 beads of each color. Now, out of this 48, you further divide 24, 24. So, 24 of one color in one beaker, you take another beaker. So, 24 of one color in one beaker and 24 of the same color in other beaker. That means, all the 4 color beads you have 24 in one beaker and 24 in other beaker. So, 24 into 4 you have in one beaker and 24 into 4 you have in second beaker. You can name them beaker A, beaker B, and both are having identical beads, colors and numbers. Now, because we are working on dihybrid cross, so we have to join 2 characters. We can't move single bead onwards. So, now you will join the beads in a particular proportion. We will put beads into pairs in a particular color combinations. For example, if we have taken red, yellow and black and green beads, in that case we will do the pairing of red, yellow, then red, black, green, yellow and green, black. For making the pairs, we will need needle and thread, which children you should keep ready. If you are doing this experiment, you should have needles you should have thread and of course scissors. And the way I said that we have to make 4 different kinds of pairing of beads, that way you make pairs, like I have taken out one white and one red. Now these 2 beads I will put together. So this is white one, it has come into thread. Now I will put red, it has also come in thread. Now I will tie it so that it is firmly together. It should not separate when we are conducting the experiment. I will give at least 2 knots. One knot, one more I will give, otherwise it may open up. Now pair is made. I will cut the extra thread. So this is our one pair, this is another pair. Like this we will go on making pairs as the color of the beads prescribed, and we will have 4 different kinds of pairs which I made some and kept ready. These all are pairs with different kinds of colors of beads. This is black and white. After you have made the combinations, now our work begins. So you have these bead pairs in 2 beakers now. You will exactly count and put. I would like to remind you children that we have made 4 different combinations of these pairs, and these combinations I repeat: red and yellow, red and black. That means you had 24 red beads, 6 will go with yellow, 6 will go with black. Similarly, I am talking of one beaker now, green and yellow, green and black, 6 6. This is one beaker and similarly the second beaker. And after you have done the pairing, then you are putting them in 2 different beakers. Now the main experiment begins. Keep these 2 containers, maybe beaker, maybe petri dish, maybe watch glass, and without seeing you will pick one pair from one beaker or one container and other pair from other. That means you have 2 pairs. By chance both are black and white. Now how you have to cluster them? You will cluster them red and yellow, then red and yellow and red and black. Why I am saying 2 colors? Because you have 2 beads in every pair. So red yellow and red yellow, if it comes together you will put in one cluster. Red yellow with red black other one, then put another cluster. Green yellow with green yellow, if it comes, you do not have to select and put, and green yellow and green black, because these 4 are the combinations which you are going to get. So now you have 4 clusters of these 4 different pairing. So every time you are taking one pair from here, one pair from here, without seeing and then see what it is. Now, red black, by chance both are again identical. So, you will make another cluster. Again you will take one from here and one from here, and please do not look into the beaker. Now, suppose two pairs come, then drop it back and take single. Now, you have one black red and one white black. So, accordingly it will go in different clusters. What I mean to say, children, you are making 4 clusters of dominant and recessive characters taking into consideration. So, in this case it is red yellow in one, red yellow, red black in one, green yellow in one, and green yellow and green black in one. Now, when these 4 clusters were made by Mendel, to his surprise, the ratio was 9:3:3:1, and any number of times he repeated the experiment, he got the same ratio. He tried to analyze how it happened. It was the case of 2 characters together. In this you can see the gametes are left side female gamete, capital R, capital Y means both are dominating characters, small r, capital Y, one dominating, one recessive. Similarly, capital R, small y, again another dominating and another recessive, and small r, small y, both recessive, it is female gamete, and similarly you see towards the right side the male gametes also. Now, when you put them 4 on this side, 4 on the other side, you can make combinations in the square which are having 16 individuals, the offspring you can say. So, capital R, capital Y on one side, capital R, capital Y on the other side at the number 1, it will make the combination capital R, capital R, capital Y, that is dominating for both the characters, that is round seeds and green seeds, the round seeds which are green. Similarly, when you come small r and capital Y, then other side also you take small r and capital Y, you have different combinations. When you calculate the total, you will find that there is one fully recessive which is shown on the lower side, it is fully recessive, both the characters recessive, that means wrinkled and yellow seeds. So, children, in this dihybrid cross, Mendel observed that out of 16 plants, the ratio was 9:3:3:1. That means 9 were having fully dominating characters, round seeds and green seeds, and 1 was having purely recessive character, that is wrinkled and yellow seeds, whereas 3 were having, you can see in this diagram also, 3 were having not round seeds, but wrinkled seeds, but green in color. Similarly, 3 were having yellow seeds with, so yellow seeds with round seeds. With the result, these 2 were hybrids. You can see here, this one is pure recessive with wrinkled yellow seeds. This one is, that is 3, 9:3:3:1 hybrid. Small r means wrinkled and capital Y means green. So, green and wrinkled 3, and similarly another 3 ratio was this, where it has round, the dominating character, round seed, but it was yellow in color. So, in this cross 9 out of 16 were green and round, both dominating. This was not round, wrinkled and green. Hybrid, that was 3, 9:3. Now, 9:3:3, this one round seeds with yellow color, and this one which is only 1 wrinkled seeds and yellow seeds. So, ratio became 9:3:3:1, and that was a permanent ratio for dihybrid cross. That means Mendel's experiment was confirmed. When he did it with 3 characters, again he reached to some definite conclusions, 4 characters, but after that counting became very difficult. So, Mendel proved in all possible ways that his laws are correct, and when you take one character into consideration, then it is 3:1 phenotypic and 1:2:1 genotypic. When you take 2 characters into consideration, it is 9:3:3:1 phenotypic. Children, of course, I have told you that how you will make the pairs with thread and needle, but there are other ways of making pairs also. You can just put 2 beads together with the help of glue or any sticking material, but in that case you will not be able to separate it afterwards. Or you can use stapler pins, put the pins and press them so they are together, or you can use even very small safety pins. So, there may be variety of things which you can use in making the pairs. Of course, I have used the beads. I am considering bead as one gene, but you can also use some other material, maybe seeds, maybe some dal, even for monohybrid and for dihybrid also. Only thing, the item should be identical, like you can take black chana and white chana, or you can take 2 kinds of rajma, or you can take say sabat moong, whole moong and whole urad, one is black in color, one is green in color. You can take some buttons of the same size, but different colors. So, variety of things can be used for this experiment, whatever suits you, but characters in dihybrid cross are to be joined. So, you can join either with thread or with glue or with pin or with any other mean which you can think of. That is one thing. And after that you conduct the experiment several times and take the average for accuracy. So, in today's session we have discussed about dominance, incomplete dominance, taking example of dog flower where red and white flowers gave you pink flowers, but eventually it followed Mendel's law in the F2 generation. We discussed codominance where 2 characters are shown together and we gave the example blood group in our body. And then we have discussed dihybrid cross given by Mendel, and we did it practically taking help of beads, making pairs of the beads. That means characters are paired and then picking up the characters without seeing and making the clusters and counting the ratio which came to 9:3:3:1. So, this shows the characters move independently and there is independent assortment of characters. With this we come to the end of this session. Thank you.